Doing another video for A-level biology year 1 or year 12, focusing on viruses, and obviously looks at the HIV virus as the main one you have to know about. So what do we need to know about viruses? Well, you need to know that viruses are acellular and non-living. So what that means is technically they're not a proper cell because they don't have a cell membrane, and they are non-living because they only reproduce when they're inside another organism. So and they've got no means of independent growth or independent metabolism. So they have to have a host to evolve and to develop. So therefore, it's questionable whether they are living or not. How do they get into the cells? Well, it depends on what type of cell they're actually trying to get into. So if they're trying to get into a plant cell, they have to tear through the cellulose cell wall. If they're trying to get into a bacteria cell, they'll have to punch a hole and inject the DNA through the cell wall of the bacterial cell, which, as you remember, is made out of peptide glycan. And if they're trying to get into animal cells, they'll normally get into animal cells via injecting themselves into it, okay? Or they'll go in via endocytosis. When the cell engulfs bacteria, they'll go in attached almost to that bacteria. So the virus that we need to know about, obviously, is HIV, which stands for Human Immunodeficiency Syndrome. So relatively new virus in terms of our knowledge of it, because it only really appeared in the early 1980s, and it's a very complicated virus. So overall, it's got, as you can see in these two diagrams, it's got a spherical shape, so it's like a ball effectively. On the outside, it's got a coat of lipids and glycoproteins, so it almost looks a bit like you think about the COVID-19 virus, which we all know about now. But it is obviously a different structure on the surface. Inside, there is a capsule that contains RNA and an enzyme, really importantly, called reverse transcriptase. So we have a look at this structure here. You can see overall it is a lipid. So this area is a lipid membrane, as it sort of says here, with glycolipids obviously on the outside. We've got the RNA on the inside, and as I said, that is all in a capsid, which will again be made of most likely be fats and stuff like that as well on the inside. So HIV then, how does the virus spread? Well, as you know, it only spread from person to person. If you're infected, obviously you can pass it to other people via unprotected sexual intercourse, by blood transfusions from an infected person, or if you're sharing needles, if you're taking drugs, that could come from an infected person there. And obviously it could come on through blood transfusions or organ transplants, but obviously we won't generally have that happen these days because everyone gets tested really well before you obviously give blood. You can't get it from kissing, hugging, sharing food, insect bites, toilets, bathing together, sneezing or coughing or sweating. So it's not a virus that can be passed like that. But it's only those 3 ways which it can be passed through. So how does it actually cause the disease? How does it spread? Well, the key thing about HIV, which is why it is such a dangerous virus for those people that catch it, is it infects your immune system. So the cells that are trying to fight off disease in your body are the ones that are affected. And it affects the T cells, and in particular the T helper cells. So if you remember from the previous video or so, we talked about T helper cells. One of the jobs of T helper cells is it stimulates the beta cells to start doing clonal expansion, clonal selection, to make those antibodies. So in order to make, remember, immunity really is basically the making of your antibodies to fight off disease. If your T helper cells are damaged, that then triggers a lot of problems in your T cells, and it triggers a lot of problems in the B cells being made. So your immune system immediately starts to drop. So what happens is it infects the T helper cells, and it will inject the viral messenger of viral RNA into the cell, and then it will use the enzyme reverse transcriptase. And the enzyme reverse transcriptase allows the conversion of RNA into DNA. So it turns its RNA into viral DNA, makes it double-stranded, and then it inserts it into the hosts, which is obviously normally a human in this case, DNA. Then as the host or as the cell, the T cell starts replicating, the viral DNA starts replicating with it, okay, and it divides, making lots and lots of copies, okay. So the viral DNA gets copied, but it obviously doesn't actually have an effect on you at the minute, and you are HIV positive. So if we continue with that then, so this is what then happens. So if you have a look at these pictures at the bottom, it sort of shows you the stages, what you'd see in a blood test. So if you were not HIV positive, you would have just the blood cells there. And then after the point of infection, you go through several stages. So first of all, you would have acute HIV infection, which would be like the flu effectively. And that is because initially the viral infection replicates rapidly and damages several of your cells, and therefore your body detects that there's something wrong, and it starts producing as many antibodies as it possibly can to destroy the HIV virus. And that's why when you're HIV positive, you will have HIV antibodies inside your body, which links back to the ELISA test from previous lesson. However, because the HIV virus is hiding in your T helper cells, it doesn't get destroyed. So then what will happen is it will spend that time hiding in your T helper cells, and it will, do very, very little replication. It will be replicating as these years go by, but not enough to cause a massive immune response in your body, and they quickly infect other cells. So there'll be some getting destroyed, some not getting destroyed. But over the years, people get their basically HIV virus levels monitored, and at some point though it becomes AIDS, and that so we go from this latency period when the replication drops and you get no symptoms, you live a relatively healthy life, normal healthy life, but then the immune system will start to fail when you get acquired immunodeficiency syndrome or AIDS, and that is when your T helper cells, the count starts to drop because they're getting too damaged and your immune system gets too exhausted, and therefore it drops below a certain level, and then the HIV virus spreads rapidly into your bloodstream because your immune defense has gone. Now, when you get AIDS, obviously that's when it becomes a problem. So HIV becomes, after the latent period, becomes active and it takes over the cell. More HIV viruses are released, killing that cell, and then releasing thousands of new viruses into your bloodstream, which will kill further cells. These infect new T helper cells, and the body's T helper cells are then destroyed. And you've got no T helper cells, so then beta cells can't be activated and you can't get any antibodies. So it starts becoming a vicious sort of circle. And you start to get, first of all, opportunistic diseases. So a lot of people who have HIV die from other things. They don't die from AIDS. They die from opportunistic diseases that affect them. So something called Kaposi's sarcoma, which is a type of cancer that people get often because your cells are exhausted. They can't pick up mutations. Tuberculosis, pneumonia, TB is a common one. So if we have a look then at the levels of it, then first thing what happens when you have acquired immunodeficiency syndrome or AIDS, and it goes from this point where your body's been coping, so now you've got too few T helper cells. The first thing you'll start to get is minor mucous membrane infections. So you mean like lung infections or possibly like digestive infections, things like that. So where there's thin mucous membranes, that is where it might happen. And reoccurring respiratory infections, so again, lung infections, chest infections are common. Then the immune system continues to deteriorate, so then you might lead to having chronic diarrhea, severe bacterial infections, such as TB, and that's obviously a key issue. So if you remember, we've said something like 2/3 of the world population have TB, but only 10% of it ever becomes active. Well, HIV, obviously, the immune system drops, so TB can become active and it can really harm people, obviously kill people. And then your low immune cell numbers continue to drop, and that then causes particular diseases, so something called toxoplasmosis of the brain, which is parasite infection, candidiasis, I can't really say that, which is a infection in the respiratory system. So obviously not particularly pleasant. Now, in terms of treating HIV, obviously there is no cure, but the drugs are pretty good now, to the point where if you've got the money to be able to take them, and obviously in this country that would be NHS prescribed, but in other countries it obviously isn't, you could slow down the replication of the virus to have a pretty normal life expectancy. Antibiotics obviously can't kill the viral infections because they don't have the cell wall and they can't be interfered with their metabolism like they can with bacteria. So on the slide here, we've got all the different drugs that are associated with HIV. So it shows a very complicated So if we have a look at some of these methods that we can use using these drugs, it shows you how they sort of work and how they sort of fight off the virus and slow it down, obviously not killing it. So we've got here reverse transcriptase inhibitors, which means stops obviously that RNA being converted into DNA before it can incorporate it into our cells. Down here you've got protease inhibitors, and protease inhibitors are obviously going to stop the virus being able to make new proteins to move from cell to cell. And then you've got entry inhibitors, which again are going to stop you getting that virus getting into your cells as a result, and therefore stop that increase in those virus numbers. And all these things obviously extend that latent period where you have no infection rate. Obviously, there have been a few cases, if you watch the news, where people now they're suggesting some drugs are starting to maybe even potentially kill the virus. So that's really good news, but obviously they're probably a long way off for that being widespread. So there is no absolute way of killing it at the moment and curing people with HIV. So the main reason that we need to know about HIV is we need to understand it, and obviously therefore looking at what we did last time, it's about ELISA testing. The indirect ELISA test allows people to know if they've got HIV virus if they suspect they have, and then that can stop people then having unprotected sex. Which will mean that they're using condoms, which means that they won't pass on the virus. And even if you don't know, obviously that is a really useful way of protecting yourself against HIV or other sexually transmitted diseases. Also using needles, if people take drugs, it's about using sterilized needles and not sharing needles. And also that last one, obviously, if you're pregnant, you can take drugs, and that stops the HIV virus being able to get across the placenta into the unborn baby's bloodstream. So that's been useful, guys, and I'll see you again soon.